going to be about some basics that one should know when we are dealing with cancer. So the topic of the lecture is Pathophysiology and Molecular Biology of Cancer Processes. And this is the outline of the lecture. I will talk about some cell cycle and its regulators. Then we will touch upon tumor suppressor genes and oncogenes, and then DNA repair pathways. There's a lot of things that I've put together in this lecture, but I hope that by the end of the lecture, we'll be able to make some sense out of it. So cancer, as we know, is a disease of uncontrolled cell division, and its development and progression are linked to a series of changes in the activity of cell cycle regulators. And that's why it's very important to know the basics of the cell cycle, and so that we can, use this knowledge to somehow in the therapeutics of the cancer. So the basic function of the cell cycle is to duplicate the DNA in the chromosomes and then segregate these copies precisely into two genetically identical daughter cells. And this is how a normal cell cycle looks like. So there are main two phases of the cell cycle, as you can see here. This is the M phase, which we call M phase because it is the mitosis where the chromosomes divide. This cell basically, mother cell divides into two daughter cells. And then one is the S phase, which is where the DNA replication takes place. The M phase is the M phase, and the rest of the phases are called the interphase. So normally between these two main phases, that is the synthesis phase where the DNA duplicates and the mitosis, there are gap phases which are called gap 1 and gap 2. So when we read in the textbook, it usually says that the normal cell cycle has 4 phases, which is G1, S for synthesis phase, G2 phase, which is the gap 2 phase, and the mitosis phase. And what happens, we'll talk about what happens in these different phases of the cell cycle. So gap 1 phase is basically characterized by, there's a lot of biosynthesis of the proteins and the organelles that are required for the synthesis of the DNA in the next phase of the cell cycle. So basically, in this phase of the cell cycle, the cell grows, and there is a growth and production of the proteins that are required for DNA synthesis. And the duration of this phase is highly variable. Usually the smallest phase of the cell, the whole cell cycle, is mitosis, where the actual division of the mother cell takes place, which lasts for only 1 to 2 hours. And then the rest is taken up by the interphase of the cells. Then G1 phase, which is a very important phase, and we'll come to it why it is very important, it varies from 5 to 9 hours. This is then the S phase, which could vary from 6 to 9 hours, and then the G2 phase, which can vary from 2 to 5 hours. In the S phase, there's a DNA that is double, so the DNA replication takes place in the S phase, so the so-called the synthesis phase, and then the chromosomes have to replicate into the sister chromatid. And in the G2 phase, basically, there is the preparation for the final cell division. So there are a number of different proteins that are required for the actual mitosis of the cells. So the mother cell has to divide into two daughter cells. So that the preparation for that phase is where the G2 phase is, and the chromosome consists of 2 chromatids. Sorry, I have to go back. Previous. So then, like we saw that there are multiple phases of the cell cycle, and as you can assume that we these phases have particular function in a cell cycle, and it's very important to control these functions because we do not want uncontrolled cell division. So there are 2 main components of a cell which actually regulate this division of the cells. And these 2 components are cell cycle that control the cell cycle, basically, are cyclins and cyclin-dependent kinases. So kinases, as the name suggests, these are the proteins that ultimately lead to phosphorylation of the downstream signaling proteins that would the effector proteins that would then eventually lead to either the synthesis of the DNA or the division of the cells or all the downstream processes. But these are not active in the cells where the cell is when the cell is resting, let's say. Only when the cyclins bind to these CDKs, the CDKs become active, and then they phosphorylate their downstream proteins. The phosphorylation of a protein can have two functions depending upon what kind of protein it is. Phosphorylation can lead to inactivation of a protein, and phosphorylation can also lead to activation of a protein. So depending upon what there are different types of CDKs and cyclins, and what type of downstream protein they are binding to will lead to either inactivation of a protein or activation of the protein. So cyclins, so the name cyclin is because these proteins, they oscillate, as you can see over here, between different phases of the cell cycle. So there are very specific cyclins that are expressed at a certain phase of a cell cycle. And then once that phase is gone, these cyclins have to be degraded, and the newer cyclin that are required for the next phase of the cell cycles are expressed in a cell. So they kind of oscillate during the cell cycle, and this is mediated by, so the degradation of these cyclins is mediated by, it's a proteasomal degradation. So there are some ubiquitin moieties which target these, which binds to these proteins and then target these proteins to proteasomal degradation in a cell. So cyclins, that's how the name cyclins come from. They are among the most important core cell cycle regulators, and they bind to CDK, like I mentioned before, to regulate their activities. There are mainly 4 basic types that are found in humans. G1 cyclin, which promote the passage through the start or the restriction point in late G1. And this start, which is in a yeast, and the restriction point, will come to it later, because these are very important phases of the cell cycle where actually there's a lot of, what should I say? Lot of commitments that a cell make to divide further are made. So, G1 cyclins, they promote passage through this. Then there is G1S cyclins, which bind to CDKs at the end of G1, and at that point of time, the cell is committed for DNA replication. S cyclins, they bind to CDKs during S phase, and they are required for the initiation of DNA replication. Then there are M cyclins, and these promotes the events of mitosis. So mitotic cyclins, they accumulate gradually during the G2 phase of cell cycle, which precedes the M phase, where I mentioned before that there is a lot of preparation for the M phase. So they gradually accumulate, and once they reach a concentration which is high enough to bind to the CDKs, then that complex is called mitosis promoting factor, which then leads to that cell into the next phase of cell cycle, and the cells enter into mitosis. The other important protein, like I mentioned before, are the cyclin-dependent kinases, Cdks. So Cdks are kinases that phosphorylate many intracellular proteins that initiate or regulate the major events of the cell cycle, like DNA replication, mitosis, or cytokinases. And how this is an example of when the cell has to move from G1 phase here to the S phase, and how these processes are mediated. As shown over here, there are 2 cyclins, one cyclin, cyclin D and CDK4. Like I mentioned, there are many cyclins and many CDKs, and their combination so there are specific cyclins and CDKs that are expressed at different phases of the cell cycle. And CDK4 and cyclin D, when they come together, the CDK can phosphorylate a protein called RB. RB, actually, this is a phase when RB is bound to E2F. E2F is a transcription factor, which in a resting cell should be inactive, because when it's active, it leads to the expression of the cells that are required for the synthesis of DNA. So if the cell is not committed to divide, this E2F should be kept inactive. And how it's kept inactive is by its binding to RB protein. So now when the cell has to enter from G1 to S phase, the RB gets phosphorylated. Once this phospho group is attached to RB, it dissociates from E2F. And now E2F is free to basically synthesize all the genes that are necessary for the active for the synthesis of the DNA. And that's how then the cell enters the S phase. So this is how the Cdks and cyclins actually regulate transition from one phase to the next. So there are different combinations of Cdks and cyclins that are made at different phases of the cell cycle and then lead to the transition to the next phase. Here, so Cdks and cyclins, they are proteins that come together, but some signal has to control their expression as well. So here, it's a diagram that shows many different family of proteins that actually control the expression of cyclins and Cdks. Like for example, Inc family of proteins, that is P15, P16, P18, P19, they control the levels of cyclin D and CDK4 and 6. And then the next, like KIP family proteins, P21, you must have heard about P21. When P21 is active, is expressed, the cell is halted in one particular phase of cell cycle.